Hey guys, so I wanted to review DNA replication for you before your test. Uh, the process of DNA replication is actually pretty basic. I think once you get your mind wrapped around some of the players involved and some of the definitions of each, it gets much easier. Okay, so now that we know about DNA, the basis of it, what it's made out of, uh, the cycle, part of the cell cycle, this replication is going to occur, and I think things are kind of put into context much better. Uh, so hang with me for 1 second while I review what the semi-conservative model is. So you recall if you have a molecule of DNA is double-stranded. This molecule that we're starting off with would be the parental DNA. Okay. Now, if you were to denature that DNA sample, denature meaning when you apply heat to it, basically you separate these two strands, you break the hydrogen bonds. Uh, you break the hydrogen bonds between those two strands, you're going to get separate parental strands. These parental strands are both going to serve as templates. This parental strand right here is the template for this newly forming daughter strand, and this parent strand right here is serving as the template for this newly forming daughter strand, and you're going to get two separate new DNA molecules from that. Okay, so you denatured the structure, basically apply heat, it will separate itself because those hydrogen bonds will break, and then in will come the nucleotides that are complementary. So A will come with T, T will match with A, G will match with C, C will match with G, and so on and so forth until you basically get two newly formed uh, identical daughter DNA molecules. So now they're identical, but they went through a different process to be replicated. For this daughter strand here, the new part was this strand, and for this daughter strand here, the new part was here. So they used a different template, but because that template was from the same original parental molecule, the DNA strands are the same. And it should be the same, right? Because we're trying to replicate it. We're not trying to introduce any sort of new mutations or changes to the genetic material. That would actually be bad. We're just trying to replicate it. And again, this is occurring in S phase of interphase, S phase of interphase of the cell cycle. Now, just thinking about the process overall, if you have a parental DNA molecule here, uh, basically you're going to take that double-stranded molecule, but to do that you need to basically sort of pry open this strand here and this strand here. So this is what we're seeing here with these origins of replications. Now keep in mind the fact that this eukaryotic DNA is actually quite complex. So this isn't a prokaryote where we make a single origin replication. For eukaryotes, we actually make many. So you see 3 here. There are probably hundreds to thousands of them per DNA strand. This replication bubble will open up at the origin of replication, and basically what you're going to do is at this origin you're going to start to make newly synthesized daughter strands. So that's the gray shown right here. Okay, these strands basically proceed in either direction, making that bubble bigger and wider till eventually these strands are going to connect here and you'll get a single strand. Okay, so eventually these strands here will connect and that'll be turn into a single new strand. Okay, um, meaning you'll have a completely new DNA molecule in addition to that newly synthesized daughter strand. Okay, so the actual process of DNA replication. Uh, Again, you just kind of have to learn some of the players involved in this process. So you can view DNA as a zipper. I think this comparison is apt. So if this is your origin replication here, you're forming a bubble here. Basically, what you're going to do is you're going to proceed in different directions based on the nature of the DNA molecule. Okay, so you have to remember that the DNA molecule basically is anti-parallel, it's complementary. So for this top strand, you're going from 5' to 3' this way, and for this strand down here, you're going to proceed from 5' here to 3' this way. Okay, so they're anti-parallel, they're running in different directions. This, this means, though, that we have to synthesize the new DNA strands for each new daughter strand differently, depending on which side of the zipper we're on. Okay, we made it pretty clear in class that you cannot start a new daughter strand on a DNA template without this primer being laid. Okay, so this RNA primer is essentially the starting point for DNA replication. You need to lay that RNA primer down in order to proceed, and that RNA primer is laid down by an enzyme called Polymerase. Remember, it's ending in ASE here. The other really critical thing is that new nucleotides can only be added to the 3' end of a growing daughter strand. Okay, so if this parent strand up here is proceeding in the 5' to 3' direction this way, that means that the new primer here is going to be proceeding in the 5'. Okay, it's going to be proceeding in the 5' to 3' direction this way. So you're only going to be able to add here. You're only going to be able to add here, 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 and so forth in the 3' direction. You can only add in the 3' direction. The opposite is going to be true now for the other side. So for the other side, if you put down a primer, say right here, color that in so it looks similar, this would be the 5' end, and this would be the 3' end, and basically can only add here, here, here. So we're going to go this direction on this strand, and this direction on this strand. I know it sounds complicated. If that didn't make sense to you, pause it and play it back. You can only add to the 3' end of a growing, newly synthesized daughter strand. Okay. The enzyme that is adding these new nucleotides, okay, is called DNA polymerase. Okay, and we'll talk about at the end the differences between some of the DNA polymerase, but just know for now that DNA polymerase is the enzyme that's adding the new nucleotides. Okay, two other protein players I want to introduce you to before we move on. This guy helicase, basically, if you recall DNA being a twisted ladder, essentially, it needs to be unwound so that we can have access to it, and helicase is the one that unwinds the helix, basically. Topoisomerase is an enzyme that relieves stress on the distal end of the DNA molecule. So you can imagine if we're opening the DNA this way, there's going to create a lot of tension this downstream on the DNA molecule. It's like when you're playing with a piece of rope. If you pull that rope open, you put tension downstream in the rope. So topoisomerase is an enzyme that can relieve that tension. It basically breaks the DNA molecule, unravels it a little bit, and then puts it back together to relieve that tension so that we can proceed with DNA replication, so we can proceed essentially into this replication fork. Okay. Just a quick note again to see DNA polymerase in a different way. We talked in class about nucleotides. This picture shows a nucleoside. The only difference in that, so a nucleotide is composed of a nitrogen-containing base, a sugar, and a phosphate. So a nucleoside basically provides its own energy for being added to this 3' end, so to that hydroxyl end. The energy is stored, much like ATP. It's essentially the same molecule. In these 3 phosphates, right? So a nucleoside would provide the energy needed to couple itself to this growing strand. Basically, you would cleave that last phosphate off, and that would give you the energy to do that. So that's the difference between nucleotide and nucleoside. Okay. The way that a leading and lagging strand is replicated is totally different, just due to the nature of the DNA molecule. If you look at a leading strand, it's actually rather easy, right? Because you have a parental strand that's going from 5' down to 3'. So if you put your RNA primer and proceed in this direction, you're adding onto this 3' end, right? Because it has to go from 5' to 3'. You always add at the end in this way. So that's what we're doing here. We're catching up to DNA polymerase down here, and it's proceeding basically into this replication fork. It's proceeding into essentially the gap of this newly opened DNA molecule. And it does this freely. It has no roadblocks. So if you can see in this picture over here, it's really small. You start here and you basically proceed into that replication fork. You're only limited by the time with which it takes to open this replication fork here. Okay, so you proceed directly into that fork, and you just keep adding nucleotides until you reach to the end of the molecule, or until you reach another polymerase doing its thing on the other side. Lagging strands are actually quite different. When you think about a lagging strand here, a lagging strand is inhibited by the fact that this replication fork only opens so far. Okay, so if you have to lay down a primer here and proceed in one direction, you'll have to go back and basically backtrack and fill in that DNA once this opens. So you see right here, this double helix is not open yet, so we can't perform replication there. We have to wait until that opens up so we can go back and backfill the new nucleotides. Let's take a look at that in a little more detail. So you have your first round of replication here. You essentially lay an RNA primer and you proceed in the 5' to 3' direction, only adding at the 3' end. So you're constantly adding here, you're constantly adding here, you're constantly adding here. This thing occurs until it runs into another polymerase, basically. Okay, so that's shown here. Alright, so it's run into another polymerase doing its work. It runs into that RNA primer, basically. So this little Okazaki fragment, this space right here, is now complete. Okay, polymerase is free to go off and to start another Okazaki fragment, or it can go and start another leading strand on the other side. Polymerases aren't assigned to a certain strand. But let's say this polymerase goes back here and wants to start filling in another gap of DNA. Well, it'd wait for primase to set up a primer there, and then it basically proceed in the same direction from 5' to 3', only adding at the 3' end until it hits that other Okazaki fragment. So I hope you can appreciate that basically with the lagging strand, you're limited by the opening of the replication fork. You put a primer down, you proceed, you fill that space, and you proceed to the next Okazaki fragment. Now there's two interesting points here that I want to show you. It's a really big deal that we have these RNA primers still laid down. We need to get rid of them. Our DNA polymerase 3 is the enzyme that lays down nucleotides. DNA polymerase 1 is a polymerase that goes back and removes these RNA primers. Okay, so it gets rid of the RNA nucleotides and exchanges it for DNA, right? Because you want your DNA to be composed of strictly DNA. Okay, so that's actually a really important thing. The next big deal is ligase. So if you remember, we have these little kind of chinks in the strand of DNA here left by the Okazaki fragments. There is an enzyme called ligase that goes back and basically seals these little nicks in the DNA formed by the different fragments being formed. You want your DNA to be contiguous, and if you don't, that's actually a pretty big deal. Okay, this leading lagging strand shows that you do both of them on a DNA molecule, right? So it's not that you just do leading or lagging, and they occur. A leading and lagging strand occurs on both sides. So I'm running out of time here to publish this, but essentially you proceed into the replication fork for a leading strand, lagging strand, you proceed in the opposite direction and do it kind of piecemeal. Okay, so I'd also go back and check out this summary. It's actually quite good, and I can't go over 15-minute mark, so I'll stop there. Good luck with your test, and talk to you soon.